You know, inside every single one of us, there's this incredible river of life flowing, our blood. It's carrying a nonstop story about our health, written in millions of tiny cells. But how do we actually read that story? Just look at this. This is the world inside you. It's this bustling, microscopic city of cells, and every single one has a job to do. So to really understand our health, we've got to learn their language. And maybe the most important word in that language is hemoglobin. Okay, so what is hemoglobin? Well, the easiest way to think about it is like this. It's your body's gigantic, always on the move taxi fleet. Seriously, it's the most important delivery service in your entire system, and it has one single job: picking up and dropping off oxygen.
Now, that job might sound simple, but it is absolutely crucial. Hemoglobin is the protein that lives inside your red blood cells. Its whole purpose is to grab oxygen from your lungs and hustle it out to literally everywhere else. Your brain, your muscles, right down to the tips of your toes. I mean, without that constant delivery, everything would just grind to a halt.
And this is exactly why getting the count right matters so much. When that taxi service goes wrong, you've got problems. If you have too little hemoglobin, that's anemia. It's like not having enough taxis for rush hour. Your body's tissues are just left waiting, starved for oxygen. But if you have too much, that's polycythemia, basically a massive taxi traffic jam that makes your blood too thick. It's a super delicate balance. So that brings up a pretty big question, right? How did we first figure out how to count these little oxygen taxis? Well, long before we had computers and fancy automated labs, the answer was surprisingly analog. It was this clever, hands-on solution that relied on a tool we all have: the human eye. This was called the Solly method. And it wasn't some minor technique. This was the original way to estimate hemoglobin. You know, it was the very first real attempt to put a hard number on this protein. And for its time, it was revolutionary. And the idea behind it was actually pretty elegant. A lab technician would take a small sample of blood, add some hydrochloric acid, and then just watch. The hemoglobin would react and transform into this brown compound called acid hematin. The rule was simple. The more hemoglobin you had, the darker brown the mixture would be. And this was the toolkit. I mean, check this out. It looks like something straight out of a vintage chemistry set, right? You've got your pipette for the blood, a special tube for the reaction, and then the most important part, this little comparator box with a standard brown color right in the middle to match against. So the process itself was like a very careful recipe. First, you put the acid in the tube. Then you add a very precise amount of blood. You'd wait 10 minutes for that chemical reaction to happen, and then you'd start adding distilled water, drop by drop by drop, diluting the sample until its color was a perfect match for the standard. The amount of water you added told you the result. But, and this is a big but, as ingenious as this all was, it had a massive fundamental flaw. It relied entirely on human perception. And when it comes to medical results, close enough is, well, it's just not good enough. You see, the goal was a perfect, objective color match, but the reality was messy. What looked like a perfect match to me might look a little off to you. It was completely subjective. So it really boiled down to two huge problems. First, the simple fact that everyone sees color a little bit differently. And second, that brown acid hematin compound wasn't even stable. The color would literally start to fade away, so the technician was in a race against the clock to get a reading before the sample itself changed. Clearly, science needed a better way, something more reliable, more stable, and more objective. The answer was found not in just looking at colors, but in much more advanced chemistry, the cyan-methemoglobin method. And the principle here was a total game changer. Instead of creating some unstable brown color, this new method uses a special chemical cocktail that converts all the different forms of hemoglobin into one single, incredibly stable pink-colored compound. No more fading, no more racing against time.
And this is where the process becomes truly scientific. We're not just splashing in some acid anymore. This is Drabkin's fluid, a very specific recipe of chemicals measured out to the hundredth of a gram. This is the moment we shifted from just estimating to actually measuring.
So the advantages were just massive. It measures all types of hemoglobin, which gives you a much more complete picture. The pink compound it creates is super stable. But the biggest advantage of all, it takes the subjective human eye completely out of the equation. A machine now reads the intensity of the color, giving you a precise objective number. This is the gold standard that powers all of our modern automated labs today.
So, what does a normal count actually look like? Well, this chart gives you an idea of the target ranges, which we measure in grams per deciliter. And as you can see, a healthy value isn't just one number. It actually varies depending on things like sex and even pregnancy. Doctors use these ranges to figure out if your oxygen taxi service is running nice and smooth. But here's the thing. As important as that hemoglobin number is, it's never the whole story. You should think of it as one really key piece of information in a much, much bigger puzzle.
See, when you get a blood test, you're usually getting what's called a hemogram, or a complete blood count. And from that one single vial of blood, labs can get a whole panel of different data points, and each one tells a different part of the story about your health.
Take your white blood cell count, for instance. It's measured in the very same test. These cells are the soldiers of your immune system, your defense force against infections. Their number tells a completely different but equally vital story about what's going on inside you. So, the journey from just eyeballing colors to this precise automated analysis shows you how far we've come in understanding just one single number in our blood. And it kind of leaves you with a pretty fascinating question, doesn't it? As our technology gets even more powerful, what other secrets, what other stories hidden in our blood are we about to discover next?